Hello students, welcome back to my channel Csir Buddy, and today we are going to study about fertilization in mammals, and this is the first part. It is related to mammalian development. So let's get started. First of all, fertilization. This is the overall view. Overall steps which are included in the fertilization in mammals. So very first step is approach of spermatozoa to ova. Recognition of egg and sperm. So the spermatozoa will approach to the egg or the ova. So the very first step is step number 1, that the approach. Sperm activated in female reproductive tract capacitation. So the second step is that until the sperm reaches the reproductive tract, they are kind of inactive. That is, their tail isn't moving. But as they reach the reproductive tract, there are certain events which occurs which allows this to activate, and this process of activation is known as capacitation. Sperm bind to zona pellucida, that is attachment. So zona pellucida is present in the ova and the sperm kind of attached to that zona pellucida layer. Acrosome reaction, release of acrosin and lysine. Now what will happen? Acrosome is present in the head of the sperm, and due to acrosomal reaction, this layer could be penetrated by the sperm with the help of acrosin and lysine. So the sperm acrosome releases some of the chemicals, that is acrosin and lysine, and digest this layer and penetrates into the ova. Activation of ovum start meiosis II, cortical reaction, and apomixis, fusion of pronuclei. So these are the end steps. So this was the overall view what all we have to study in this particular lecture. So let's get started with the structure of egg. So this egg is the metaphase II arrested stage. The eggs arrest itself in a metaphase II. What is metaphase II? When there is a meiosis occurring. So in meiosis there is prophase 1, and then comes the metaphase. So the egg get arrested in this metaphase, prophase 2 and metaphase 2 stage. So this is the stage where the ova gets arrested until and unless the sperm comes and there is some kind of fertilization occurring. When the fertilization starts, then again it will undergo meiosis. So I think this is clear to you all. This one layer, this is known as perivitelline space. These granules which you can see, that is black color, these are known as cortical granules. Now the layer which is the innermost layer, that is known as oolemma. The inner portion of the ovum, that is known as endoplasm, and the outer portion it is known as exoplasm. The layer which is between these two, that is the cortical granules and the perivitelline space, that is known as zona pellucida layer. Now coming further, these are some of the layers. These are cumulus layer. So the very first layer which you can see in blue, this is the cumulus layer. Then comes the corona radiata layer. Then there is a filling, that is known as hyaluronic acid. And then there are some of the protein that are known as adhesion molecule. These adhesion molecules help the ova to attach to this fimbriae. So these are adhesion proteins which are helping the ova to attach to this, so that the fimbriae could take this ova to the reproductive tract where the actual fertilization would take place. So this was all about the structure of this was all about the structure of egg. Now moving further, we will talk about the details. First, cumulus layer, and second, corona radiata. So what functions are these layers performing? Secrete progesterone hormone which provides sperms its direction. So progesterone hormone कौन secrete करता है? Cumulus layer and corona radiata, from which this sperm gets its direction. The third layer is zona pellucida. Zona pellucida is species specification, and this is a non-cellular layer. It is used to specify the species. And the fourth layer, that is innermost, it is known as oolemma. So I think all these layers and all about this egg structure is clear to you all. Now moving further, structure of a sperm. So structure of a sperm, the head is made up of acrosome which releases chemicals, that is acrosin or lysine, which will digest the layer of the ovum, right? That is the zona pellucida. This is the proximal centriole, and this is the mitochondria, which is a spiral mitochondria, and these are Csir receptor. So what are Csir receptors? Sense progesterone. प्रोजेस्टेरॉन को सेंस करने के लिए कौन आता है, कौन प्रेजेंट होता है? Csir receptor. What is progesterone? Progesterone is secreted by the cumulus layer or the corona radiata, and these Csir sense this progesterone. Proximal centriole is perpendicular to sperm axis, whereas distal centriole is horizontal. And distal centriole, there are two centrioles. One is proximal centriole and one is distal centriole, and distal centriole is required in the formation of the tail of the sperm. So I think this all diagram of a sperm is also clear to you all. Now moving further, let's talk something about fertilization. Now as we all know that this is the uterus, and here is the sperm going to enter from. So this is the region of cervix region, from where the sperms enter to the female reproductive part. Now starting from the cervix to the isthmus region, there is no activation of sperm. Sperms are not active until this. But in the IA region, after crossing this isthmus region, the sperms get activated, and in the IA region the fertilization occurs. In the ampulla region, there is a temperature difference. And what is temperature difference required for? Temperature difference is required for the fertilization to happen. So capacitation in isthmus is due to thermotaxis, that is the difference between temperature. So when there is a difference between a temperature, then only the sperm gets that signal to get activated, and activation is known as capacitation. These are the fimbriae. As the pH of vagina, as the pH of uterus is very low, that is 4.5 to 5, so the sperms and the environment becomes very acidic and the sperm die. Acidic unfavorable environment for sperm fertilization, sperm movement in female reproductive tract by uterus by prostaglandin. So prostaglandin is also an hormone. Prostaglandin produced by semen for uterus contraction, from uterus to isthmus by progesterone. So in order to reach the uterus, so here is the thing, what happens is that these are sperm which are inactive. Now how will they reach to the uterus? So they secrete, sperm are mixed with semen, and semen secretes prostaglandin. Prostaglandin will help them to reach till the uterus. After that, what will happen? It will undergo, the sperm will get activated and through the process of capacitation, and it will undergo activation, and now from the uterus to the isthmus it could easily travel, as now it is active. So what is capacitation? We will be studying about this in our next lecture, and for today this is all. So if you like this lecture, please like, share and subscribe to our channel, and if there is any doubt regarding to this lecture, so please comment, let me know in the comment section. Thank you.